Okay, it's evening, I guess. I know you all feel tired, but I guess good news is that today we will have second part of the lecture that is a little bit more biological and less chemical than the first part. So we will not have so many formulae, and I guess it will be all more about some biology. I guess we start with the part with the enzymes, at least because I have some assignments in the homework for you, and I guess it's really that biologists should know about enzymes. And if we have some more time, maybe I will return to the mechanisms of reactions and we will look at them. But let's start with with enzymes. You know that basically we are some sort of protein life form. Let's put it this way. So proteins do many different works, and of course one of the most important works that they are performing, it's basically catalyzing different biochemical reactions. So all living things, they rely on the ability of the proteins to catalyze some reactions, and these type of proteins we are calling enzymes. So here are, let's say, two examples. One of them, you see these jellyfishes, and the activity of certain enzyme is responsible for the glow of these luminescent jellyfish. So the enzyme equine catalyzes the oxidation of a compound by oxygen in the presence of calcium to release CO2 and light. So here you see the molecule, the organic molecule that is basically transformed, and we see that this jellyfish can glow. Here is another example. That's papaya, and it contains papain. That is basically a protease. The proteases are basically enzymes that catalyze, let's say, cutting proteins into smaller pieces. And the interesting thing about this sort of enzyme is that it is used sometimes as meat tenderizer. So you may look on the internet or on the YouTube, there are some sort of recipes when you are taking papaya, you are taking meat, and you are basically uh, well, waiting for you are putting this meat into that papaya, and you are waiting for some time, and it will be a little bit tenderized. So they can be, of course, extracted not only from papaya, but pineapple, fig, kiwi, and ginger are good as well. So they are available either in the original fruit or leaf, or maybe purified and powdered for the shaker, diluted in salt and sugar. So you can just buy them in form of some sort of powder. And these enzymes, they work better at a little bit higher temperature, like 60 or 70 degrees. So you may find that on the internet. One more pretty neat example, some enzymes, they may couple the catalytic activity with mechanic work. So here is a kinesin, a protein complex belonging to the class of motor proteins found in eukaryotic cells. They move along microtubules, filaments, and they are basically powered by the hydrolysis of ATP. And that's why they are type of ATPases. And here is an example of this protein working. Here's a neat video, and I guess everybody likes these sort of proteins. They are basically transporting some sort of vesicles. They may transport them from the periphery to the nucleus of the cell, or they can transport them from the nucleus somewhere. But anyway, looks pretty neat, and they are, as you might guess, quite useful. Um, so let's look once again. It's really looks really nice. I guess in some of the future lectures we may consider something pretty similar. I don't think exactly kinesin, but maybe some other proteins, for example, that help our muscles contract and so on. There are pretty similar principles that we can consider. Okay, so a few points about enzymes. The most striking characteristic of enzymes are their catalytic power and specificity, so they are pretty specific. That means that they catalyze not all the reactions, but some specific reactions. I'm telling not one reaction, but reactions, because they are not perfectly specific. So yes, they are specific, but not perfectly. So there is basically a bit of range of what they can catalyze. Catalysis takes place at a particular site of the enzyme called the active site. Nearly all known enzymes are proteins. However, proteins do not have absolute monopoly on catalysis. And there are certain catalytically active RNA molecules. They are often called ribozymes. That's an interesting part, and probably you can name me at least one famous ribozyme that you should know. Who can tell me? You should have been studying molecular biology. Did you study protein synthesis at this point, or is that still somewhere in the future?
Not yet.
Not yet.
It will be
Okay.
in the future.
Okay. So I guess you will hear. Oh, we have something in chat. Uh, Ribosome. Yes, absolutely correct. Ribosome. So in the ribosome, all the catalytic activity is performed basically by the RNA. Of course, there are certain proteins inserted into the ribosome, but you will probably learn that they are performing some sort of structural work, so they do not take part in the catalysis. Moreover, the fact that RNA is capable of performing catalysis is giving us some sort of a hint into the, let's say, theory of abiogenesis. So you probably know about the theory of abiogenesis. So like living things are coming from some sort of non-living things, and if you get deeper inside of that theory, you will know that right now we are supposing that there was a period when life looked like some sort of RNA molecules. So RNA molecules were capable of performing some sort of catalytical activities and so on and so forth, and basically the life was based on the RNA. And then in the process of the evolution, it turned out that work of keeping information was transferred to the DNA, and the work of catalyzing everything was transferred to the proteins. So RNA now looks like a little bit in between, but it is clearly possible to perform certain catalytic activity with the RNA. So yes, we can do that. But right now, it's just some sort of ribosomes are probably the sort of evolutionary remnant of that ancient times when we looked like RNA molecules somewhere. I don't know. There are pretty different theories where could these processes be performed. Okay. So yes, there are ribozymes, but right now almost all catalytic activity is performed by the proteins. Good. And once more, so enzyme catalyze reaction by stabilizing transition states. So I hope you remember from the lecture yesterday what is the transition state, and this stabilizing, it makes this the energy difference between some initial reactants and the transition state makes it lower. Let's check if I have a nice picture here. No, I don't. Okay, let me draw that, I guess. So suppose This is my reaction coordinate, and this is Gibbs free energy. And here we have our initial reactants. We start here. So here are some final results. And here we have this sort of plot that shows me how the reaction goes. So now, this point, how do we call that? That's a transition state. So this is the highest point, this is a transition state. And to get from initial reactants to some final products, we need to somehow get over this peak. If you have a reaction catalyzed with the protein, you might get something like this. The same initial state, the same final state, but lower transition state. So it's easier to get over, and you will have much more molecules that are basically capable of getting from here to there. So, so here's how it works. Remember, proteins cannot, or whatever, anything catalytical cannot make, let's say, non-spontaneous reaction into spontaneous. So initial and final states are the same. They are just lowering this sort of barrier. So that's what they do. Okay. What do I have here? Here is some sort of terminology once again. So we have different classes of enzymes. Here are 6 major classes. So here are just some words that you will probably see here and there. They come from the reactions that are catalyzed. So transferases are transferring some groups, for example, and hydrolases are performing hydrolysis reactions, and so on and so forth. So here are some examples. These numbers, I guess they are somewhere used, but I'm not sure. So there is some sort of catalogs of all these enzymes, and they have some sort of numbers there, but you might guess these numbers are pretty arbitrary, so there is no point of remembering them or something like that. And enzymes, they accelerate reactions by factors of as much as million or more. But once again, they do not change somehow initial or final state. They just help to reach the equilibrium. So we talked about chemical equilibrium on the previous lecture, well, half lecture that we had yesterday, and you remember, I hope, what is this sort of equilibrium. So they just helped to reach that equilibrium much faster. So here are some sort of rates without and with enzymes. So you see these sort of coefficients, but I don't want to get into that details. Just I want you to know that they can catalyze, well, make reaction faster, achieving this equilibrium faster by factors of millions and so on. One more interesting thing, many enzymes require cofactors for their activity. So the catalytic activity of many enzymes depends on the presence of certain small molecules termed cofactors, although the precise role varies depending on the cofactor and the enzyme. I guess we will look today at mechanism of action at least 1 or 2 enzymes, so you will probably get the idea. Generally, these cofactors, they, in some sense, they augment the repertoire that we have of 20 amino acids, and enzyme without its cofactor is referred as apoenzyme, and the complete catalytically active enzyme is called holoenzyme. So apoenzyme and cofactor gives you a holoenzyme. Why that may be interesting for you as biologists, for example, if you are trying to purify some enzyme and you are performing some sort of assay to check whether it is active or not, if you were able to purify that, it may so happen that you purify your enzyme, you are testing it, and you don't see any catalytic activity. So nothing works. How could that happen? How do you think? What can be the problem? So you purified the enzyme, you did everything correct, but it doesn't work. So no catalytic activity at all. What happened? What was lost in the process of purification? No cofactor. Yes, absolutely correct. So it may so happen that you have lost cofactor in the process of the purification and you really have the enzyme you want, but it just doesn't work because it doesn't have the cofactor. And if you add that cofactor, you will see the activity just in an instant. Cofactors can be divided into two groups, so that may be some sort of metals. Mostly you will see some sort of magnesium ion, but there are other as well, or small organic molecules, often called coenzymes. They are often derived from vitamins, and these coenzymes may be either tightly or loosely bound to the enzyme. So tightly bound are called prosthetic groups, and they will be probably connected with some sort of covalent bond. Here are some examples. So you see here a table of cofactor and enzyme that needs this cofactor. And the interesting thing, if you read through these coenzymes, you will probably recognize let's say if you have ever bought somewhere in a drugstore some sort of vitamins and you checked what's in there, so what are you going to use, these all chemicals should be pretty familiar to you, so probably you have seen them somewhere. So they are all derived from vitamins. Okay. And it's a small reminder for you from the yesterday about the free energy change. So it provides us information about the spontaneity, but not the rate of a reaction. So regarding the rate, we should consider transition state, because we should be able to go through that maximum, that local maximum, to get from our initial compounds to our products. But anyway, Gibbs free energy gives us some sort of information. So first of all, reaction may take place spontaneously only if delta G is negative. So such reactions are said to be exergonic. System is at equilibrium and no net change can take place if delta G is 0. Reaction cannot take place spontaneously if delta G is positive. Well, that's reasonable. These are called endergonic. If delta G of a reaction, the delta G of a reaction depends only on the free energy of the product's final state minus the free energy of the reactant's initial state, but independent of the molecular mechanism of the transformation. That's what I told you about state variables. So you'll remember from yesterday that in the thermodynamics we have certain things that we call state variables, and Gibbs free energy is one of them. So we had enthalpy, we had entropy, we had some more, and we have Gibbs free energy. So it depends only on the state, but not on the history, how that state was achieved. Basically, the mechanism and how did you get there is the history of the system. So it does not depend on the history, it depends on the state only. And delta G provides no information on the rate of a reaction. So it's just spontaneity or not. The rate of a reaction will depend on the energy of activation, and we use delta G double dagger basically to designate that state. Here are some sort of plots. Good. So enzymes, they accelerate the attainment of equilibrium, but do not shift position of the equilibrium. So the equilibrium position is a function only on the free energy difference between reactants and products. And here we have two reactions. Here you see what happens when we have enzyme and when we have no enzyme. Here is on the x-axis you see time, on the y-axis you see product. So if we have no enzyme, reaction goes pretty slowly. Here you see here's a break in the time axis, so this means that large amount of time passes, and at some point this plot gets here. So we have almost achieved equilibrium. So for example, our initial components transformed into the product. Here is the reaction with enzyme. It almost instantly gets to the equilibrium. But you see, if you wait long enough, you get the same result. So enzyme did not shift the equilibrium. It did not shift. It did not make more product, or I don't know, or less product, or whatever. It just helped to attain equilibrium much faster than you could get with the bare reaction. So here's the point. The next point, the difference in free energy. So here are shown differences in free energy. So here we have substrate, here we have product. And we see to get from the substrate to the product, we should go through this transition state. In the presence of some sort of catalyzer, we get some lower energy, so this is delta G double decker catalyzed, and it is easier to get from substrates to the product. But anyway, delta G for the reaction did not change, but we are just more quickly getting to the equilibrium. So in some sense, enzymes function to lower the activation energy, or let's say they facilitate the formation of the transition state. Good. What else? The formation of a enzyme substrate complex is mostly the first step in the enzymatic catalysis. I will show you some examples a little bit later, but here is the idea. So much of the catalytic power of enzymes comes from their binding and then altering the structure of a substrate to promote formation of the transition state. So here you see substrate, here you are creating some sort of transition state, and then it goes to the products. So it helps somehow to get to this transition state. So substrate mostly binds to a specific region of the enzyme called active site. Most enzymes are highly selective in the substrates they bind, and here is some example of this active site. So on the right you see the active site and some sort of chemical that will undergo chemical reaction right now. And on the left you see the enzyme itself. So you may notice that only a small part of the protein takes part in the catalysis. So everything else exists basically to position these sort of catalytic things correctly in space, and maybe do some more things like, I don't know, some allosteric regulation, something else. But here is basically an example that catalysis is performed just by a small number of amino acids that are present in the in the protein. So remember that active site may include so-called distant residues. What do I mean by that? Well, if you are synthesizing protein, it's some sort of long string, yeah, so you are synthesizing and taking that amino acids one by one, and here is the that sort of string, so you call that some sort of primary structure of the protein. And in different databases you will see proteins as these sort of strings in form of primary structure. But when you look at the protein in the 3D space, you will see that these parts of the protein, that the amino acids that are taking part in the catalysis, they are pretty far apart in the primary structure, but they are pretty close in the 3D space. So they may be quite far in your database, but in the 3D they will be close enough, and they will work together to make some sort of catalysis. So that's just some sort of reminder. And active sites, they are unique microenvironments. Water is usually excluded unless it is a reactant, and substrates are bound to enzymes by multiple weak interactions. Despite there are some enzymes that during the catalysis may bind to the substrate with covalent bonds and then cleave these bonds. I guess we will see an example of that. Here are two models. Probably you have learned at school a little bit outdated lock and key model of the enzyme substrate binding. It says that substrate and enzyme, they look like key and lock, and they should perfectly match. In practice, we expect something more like induced fit model. So your enzyme and your substrate are not perfect key and lock, but when substrate starts binding to the enzyme, enzyme may change its conformation to work better with these exact substrate. So the active site will form a shape complementary to the substrate only after the substrate has been bound, and before that it may have a bit different form. So that's a more, let's say, recent ideas. Well, in addition to the strategy involving binding energy, enzymes commonly employ one or more of the following four additional strategies. So I guess we will see a few of them just in a moment. So covalent catalysis. In covalent catalysis, the active site contains a reactive group, usually a powerful nucleophile, that becomes temporarily covalently attached as a part of substrate in the course of catalysis. Of course, then this bond will be cleaved, but at some point it may attach. General acid-base catalysis. So acid-base catalysis, we will see. I guess we will see that in some future lectures. Catalysis by approximation. Many reactions have two distinct substrates, including four classes of hydrolases, for example. In such cases, the reaction rate may be considerably enhanced by bringing the two substrates together along a single binding surface on the enzyme. So you have molecule number 1, you have molecule number 2, and the protein just bring them close together so that they may react and create molecule number 3. So and metal ion catalysis, so pretty often seen. So metal ions, they can function catalytically in several ways. So for instance, a metal ion may facilitate the formation of nucleophiles such as hydroxide ion by direct coordination. For example, zinc in carbonic anhydrase. I guess we will look at this exact example today. Alternatively, metal ion may serve as an electrophile, stabilizing a negative charge on a reaction intermediate, for example, magnesium. And finally, metal ion may serve as a bridge between enzyme and substrate, increasing the binding energy and holding the substrate in a conformation appropriate for catalysis. Almost all ATPases use that. And here are two examples for chymotrypsin. Cleaves peptide bonds selectively on the carboxyl terminal site of the large hydrophobic amino acids such as tryptophan, tyrosine, and phenylalanine, and methionine. Uses covalent catalysis, general acid-base catalysis, and I guess we will look at this example. And next example, carbonic anhydrase, is general acid-base catalysis, catalysis by approximation, and it uses metal ions. Good. Here we are. So let's check at least these two proteins. Let's check their mechanisms, and then we will go further. So here is a chymotrypsin. Of course, it's a large protein, so I'm just showing you the catalytic site of the chymotrypsin. So it's just an active site, nothing more. So here we have some sort of protein coming here. So here it is. You might guess this is what? It's peptide bond. It's peptide bond. And yes, that's a substrate. That's true. So this is a peptide bond in the substrate. So R1 and R2 just mean that there is some more molecule. Now what happens? We have this residue, that's histidine, I guess. Here we have it. And here we have, I don't know, probably serine, if I remember correctly. And now we need our diagrams with our push arrow diagrams that I asked you to draw. So here what happens. So we have CO group. You remember that carbon atom is a little bit positive. So here we have a nucleophilic attack. These electrons are used to attack this carbon atom. This hydrogen is abstracted by this histidine. I hope you remember, they didn't draw here, so that's a bad habit, but anyway. This nitrogen should have non-bonding electron pair. This non-bonding electron pair attacks hydrogen, takes this hydrogen. These electrons attack carbon atom, trying to create new bond. Now carbon atom rejects its electrons and gives them to the oxygen, and we get what we had in our lecture on the reaction mechanism that we called tetrahedral intermediate. So here it is. So here is carbon atom attached to the oxygen of the serine, I guess. Here we have our tetrahedral intermediate. You see four bonds from this carbon atom. And now this tetrahedral intermediate, if you remember from our previous lectures, they are trying they are searching, let's say, searching for some leaving group, and leaving group should leave. So in this case, leaving group is this part. So here we have electron comes back, recreates double bond. So double bond will be recreated from the oxygen. This pair of electrons creates bond with the hydrogen atom. Hydrogen returns its electrons to the nitrogen. So that non-bonding electron pair is returned right here. You can check why this thing is stable. I will leave you that, let's say, to think of that why. So you can calculate the number of pi electrons right here and to think about it. But anyway, you see that right now this part is detached and goes away. So you have this sort of thing is just going away. So here we have cleaved this bond, and now it can just this is just a hydrogen bond, so it's quite weak and it can be pushed away. Now we have this part of the peptide still covalently bound to the oxygen. What do we do with that? Water comes in and works in a pretty similar way as we had previously. So hydrogen is abstracted by the non-bonding electron pair on the nitrogen. That's good. We have this oxygen connected in here. Once again, tetrahedral intermediate. Then everything goes as in previous step, but in this this leaving group is all this part. And one more part of the initial protein goes away. So we have cleaved a peptide bond. So here you see the catalytic strategy of the chymotrypsin. What's interesting here, here is some sort of oxyanion hole. Here it's in a little bit enlarged. It's somewhere here. and it helps to stabilize the tetrahedral intermediate. So you see some sort of hydrogen bonds that help to stabilize this negative charge on the oxygen, and that's why it facilitates the creation of the intermediate state that is later used to create final products. So that basically facilitates cleavage of the peptide bond. Okay, so of course, this mechanism does not account for all characteristics of chymotrypsin, because I told you that it has a certain specificity, and it turns out there is some sort of specificity pocket of chymotrypsin. So it is lined with hydrophobic residues, and when a large peptide that should be cleaved coming to the chymotrypsin, some part of it should get into that pocket, and you should guess that only hydrophobic residues are really welcome in there. So it with this way it basically achieves certain specificity which bond to cleave and which bonds not to touch. So that's it. What else? Some note for you. So enzymes do not have preferred direction. So please remember that. That's a really crucial part. So what I told you, enzymes just help to achieve equilibrium. They do not influence equilibrium. They do not force products into. They do not force some substrates into products. They are just helping to overcome that transition state. They do not care in which direction reaction goes. So they help to achieve equilibrium, and basically if you have a state where you have a lot of products and you add enzyme, it will start synthesizing, for example, your substrates that you had. If you have a lot of substrates, you add enzymes, you start getting products. So you are just getting the equilibrium. If equilibrium is so that you have 50-50 your products and your substrates, you will get to that 50-50. It will not prefer certain direction. So okay, what else? Zinc atom. I'm not sure why did I put this in here, but okay, so zinc is found only in +2 state in biological systems. Zinc atom is essentially always bound to 4 or more ligands, and here is an example of 3 histidine imidazole rings, and an additional coordination site is occupied by water. Okay, so here is, or it may be hydroxide ion depending on pH. Okay, what's that? Here is carbonic anhydrase. So basically, I guess I was meant to show you one more mechanism of a reaction of carbonic anhydrase, how that works. So here is once more a histidine residue. Here is the zinc. So we have the zinc coordinated to some different histidine residues and to water. Now what happens? Once again, nitrogen on the histidine residue abstracts hydrogen. We will often see this sort of part of the mechanism. So yes. And what happens then? If some sort of base comes, it may transform to base to the conjugated acid, I guess. Yes. So binding to zinc lowers the pK of water from 15.7 to 7. So it becomes pretty acidic and pretty willing to basically reject one of the hydrogens and stay in the form like HO minus. So that's it. And here you see like one of these hydrogens was lost and went away with the base. And what do they do? So here is an example of water attached, and basically when attached to zinc, coordinated to zinc, it turns out pretty acidic, so it's happily rejects this hydrogen. And here is basically the example of carbonic anhydrase mechanism, how that works with this zinc atom. So here we have H2O coordinated to zinc. It rejects hydrogen atom, transforms into HO minus. Then carbon dioxide comes, so here it is. Here we have the electron pair on the oxygen. It takes carbon atom, so here this arrow. We are creating basically a new bond. Electrons from here go on to the oxygen, so we know that oxygen is electronegative, so it pretty happily takes these electrons for himself. Of course, it acquires some minus charge. This minus charge, you see, it's stabilized by zinc. So zinc has charge 2 plus, and it basically coordinates to oxygen and stabilizes that minus charge. And what do you see? What do you see here? So what's this? So what this? The part I have like encircled. How does carbonic acid look like? We have carbon atom, we have oxygen, so that's carbonic acid, yeah? Do you recognize it here? Yes, just conjugate base, so not subjugate. Subjugate is when you're dealing with your enemies, and conjugate is from conjugation. Good, yeah, so that's basically carbonic acid that has lost one of its hydrogens, so it's in a state of conjugated base. Yeah, that's pretty true. And here is the 3D example, how that looks like. So here is carbon dioxide, here is zinc. So all these metal ions, they are pretty large, and it's performing this reaction. Good. Now we have a bit more time, so it's time to get into few well, I'm not really sure to show you these equations or not. Do you feel confident with differential equations or not? Okay. No, not really. Okay, then I'll try to alleviate all these things and some sort of just dash through all this. Good. The idea is basically as follows. So we have certain chemical reactions, and as you remember in the idea of dynamic equilibrium is that something is happening, so you are getting left to right, right to left, and so on. And we can describe that source of fluxes of the components as they transform in form of, well, in form of differential equations, but I will not get into that. The main idea is basically what? So I can write the change in the amount of initial components in the form of how many of these components are transformed into some intermediate state. So here is basically E, it's enzyme, S is substrate. ES is enzyme substrate, so enzyme is bound to the substrate and they are in some sort of intermediate state. And here are enzyme and products. So enzyme has performed the reaction and we get some sort of products. The idea is that I can write some sort of flux from here to here, and it basically depends, it is proportional to the probability that I have chosen some random molecule, and that is enzyme, multiplied by the fact that it collided with another molecule that is substrate, and there is some sort of coefficient of proportionality, so K1. Of course, there is some backflow, this comes with a minus sign, and some backflow comes with a plus sign, so K minus 1. Of course, on the concentration of the enzyme substrate that we have there. And it turns out that we can collect the things and basically describe changes that we have in concentrations of all these sort of molecules in form of differential equations. Yeah, they look a little bit complicated, but never mind that. So the idea is that we can do that, and that we often get these sort of things, so some coefficient multiplied by concentrations of something, and these concentrations are basically concentrations that you have in here. And if we do that, if we suppose that if we do certain assumptions, for example, that there is almost no backflow from the product back to the enzyme substrate state, for example, it's just the beginning, so we have only enzyme and substrate, and there was no time to get a lot of product, and the probability of finding some product there is almost 0, so that's pretty okay. We do some more assumptions are that the total enzyme concentration remains constant, so the number of free enzyme and enzyme bound to the substrate is equal to the total amount that you have added at the beginning of the experiment. And assumption done with the Briggs-Haldane, it is assumed that the concentration of the enzyme substrate complex remains approximately constant during the reaction. Well, pretty reasonable assumption. So if you take all that assumptions and you do certain mathematical manipulations with all that things, basically you don't even need to solve a differential equation. It's just easier to express the ideas in forms of differential equations, but there is a pretty straightforward way to get to these equations. So this is the equation, basically the velocity of formation of the product that you have at the beginning of the reaction. So when the reaction just starts, so you have just added everything to your sample tube, and you are measuring somehow how the reaction goes. This is the expression for that initial velocity. And all in all, if you introduce Michaelis constant, so here it is, if you introduce this sort of constant and a pretty high substrate concentration, you will obtain so-called Michaelis-Menten equation. So here it is. So all the previous parts are basically some sort of assumptions that we need to formulate this equation, and here is this equation. And many enzymes, they follow so-called Michaelis-Menten kinetics. So we will probably see them in the future, and we will probably operate with this sort of thing with Michaelis constant. So it will characterize for us the enzyme and how good it basically works. Here is the plot that shows us what's happening if we are increasing the initial substrate concentration. Here is reaction velocity at the beginning of the reaction, so V0, and here is Vmax. It's the maximal velocity that can be achieved by the enzyme. And you see that when we are increasing the concentration, the enzyme is getting closer and closer to that maximal velocity. Well, that's when all the enzyme molecules are basically used up and they perform some work. And here from the equation you can basically guess the meaning of the Michaelis constant. So you see, if this Michaelis constant is equal to the substrate concentration, or rather, let's say that I have added substrate in such amount that its concentration is equal to the Michaelis constant, you will get what? So V0 equals to what? Can you tell me? No, nobody can. So I'm setting the concentration of the substrate equal to the Km. Km here and Km here. So now I can divide through and remove. Oh, I get some answer. Vmax over 2. Yes, absolutely correct. Vmax over 2. I'm dividing through here, here, and here I basically get 1. Here I basically get 1. 1 plus 1 equals 2. Knowledge bump from biochemistry course, yeah? Okay. I hope I will able to give you some more knowledge bombs. This sort, this type. Okay. The significance of Km is pretty clear if we set concentration equal to Km. So here's what we get. So V equals Vmax over 2. So Km is equal to the substrate concentration at which the reaction rate is half of its minimal value. So that gives us some idea which concentrations of the substrate you want to use with your enzyme. Here is the example of alcohol dehydrogenase and aldehyde dehydrogenases. And the thing is that most people have two forms of the aldehyde dehydrogenase. So it's this one. So alcohol dehydrogenase it transformed ethanol into acetaldehyde. That's basically the thing why you have hangover if you have drunk too much. And here is aldehyde dehydrogenase that transformed that into acetate. That's pretty nasty thing as well. So at low Km mitochondrial form worse, and in high Km cytoplasmic form. In susceptible persons, the mitochondrial enzyme is less active owing to the substitution of a single amino acid, so ALDH2 gene, and it's a common trait in East Asian people. And acetaldehyde is processed only by the cytoplasmatic enzyme because this enzyme has a high Km and achieves higher rate. So consequently less acetaldehyde is converted into acetate, and excess of the acetaldehyde escapes into blood and accounts for physiological effects like facial flushing, rapid heart rate, and ingesting, and that's even after ingesting small amounts of alcohol. And it's a common trait in East Asia, and I can assure you it's really true because I had some opportunity to work in Korea for 2 months, well, as the programmer basically, and I saw, well, yes, that's true, and they are drinking a lot. That's another knowledge bomb from course on biochemistry. Good. Here is interesting plot that you will need to solve your homework, so it's really good that we got here, and it is called a double reciprocal or Lineweaver-Burg equation. You will often see in biochemistry or in biology in general when people are creating some sort of equation and then they are transforming this equation into a form where they can do something with measurable parameters to get a linear equation. So this thing is basically Michaelis-Menten equation just transformed into a different form. So if you play around with the Michaelis-Menten equation that we had, you will be probably able to transform it into this. What's interesting here? Let me show you. We have this thing and this thing. So the result, 1 over v naught, so the reciprocal of the initial velocity, depends linearly from the reciprocal of the substrate concentration. So if you change the concentration of the substrate and you measure the initial velocity, so these are measurable things, so you know how much of your substrate you have added to your test tube, so you just know that, you know what you do, and you have measured somehow these velocity, well, different methods, so you have somehow measured how quickly your products appear, so it may be by, I don't know, changing color or something else, many methods. You can get points, and all these points should lie on a line. So that's basically the reason why everyone likes this form of equations, because we know how to draw line through points. So drawing something more complicated, some parabola or whatever, is, well, a bit more complicated. But drawing straight line, that's easy. And if you have this line, you can basically get your Km and Vmax. So the intercept, here it is. So intercept. So that's the point when where your line intersects with the y-axis. So here is y-axis. This is 0. This y-axis. So the point where it intersects the y-axis is 1 over Vmax. You will have a homework where you get these sort of data, concentrations and V naughts, and you will need to calculate parameters of Vmax. So here is at least one working method for you. So you calculate reciprocals, you draw a line through that points, and you look where that line intersects y-axis. And that will be 1 over Vmax, so you can calculate Vmax from that. Another interesting point is here, where this line intersects the x-axis. This is minus 1 over Km. So you draw a line, you look where is this point, and you look what is the value here, so I don't know, let's say minus 5, and then you calculate your Km should be equal to what? 1/5, yeah? Good. Any questions so far? No, good. So Km values, they range widely, and for most enzymes Km lies between 10 to the minus 1 to 10 to the minus 7 mole per liter. So that depends on many things, including pH, temperature, ionic strength, so on and so forth. So here are some typical Km values for different enzymes. You may want to check that. Here is some link for you. Let's check out what do I have here for you, and and then here it is. So here is an example of different well, they are basically here is the uninhibited version. So here is a Lineberg plot of certain enzyme, and you see how that, so just a straight line. And if we plot a Michaelis-Menten plot, so that was the plot of reciprocals, and here is a plot of non-reciprocals, so just substrate concentration versus the rate of formation of the products. I guess we will return to these plots in a moment, because I need to show you the effect of inhibition on different proteins. Good. Not to forget, so not all enzymes obey Michaelis-Menten kinetics. So there is important groups that falls off. They are called allosteric enzymes. So we have seen one of the examples of these enzymes. Third lecture, hemoglobin. So I was talking something about hemoglobin, and I definitely remember I showed you these sort of sigmoid shaped plots for hemoglobin, and I should have told you that this is an allosteric enzyme. So these enzymes often consist of multiple subunits and multiple active sites, and they often display sigmoidal plots with the reaction velocity V0 versus concentration S. In allosteric enzymes, the binding of substrate to one active site can alter the properties of other active sites in the same enzyme molecule. So we had a cooperative binding in case of hemoglobin, so binding to one site made more probable binding in different sites. Good. Okay, here are some sort of examples with inhibition. So that will be part of your homework as well. So I think we need to get through all of this. Okay, so example number 1. So no inhibition, pure Michaelis-Menten kinetics. So here is Michaelis-Menten equation. Here is Lineweaver-Burke equation. And on the right, you see Lineweaver-Burke plot and Michaelis-Menten plot. So nothing unusual. We have seen that. Where to pay attention? So this scheme. So the logic of the process. We have enzyme plus substrate. They transform into some intermediate state, and then you get enzyme plus product. What happens if we inhibit? Well, there are two types, basically two branches of inhibition. Irreversible, so when some molecule inhibits your protein binding quite tightly, so it may be covalent, non-covalent bond anyway, but it does not want to dissociate. Or reversible, so it turns out it can rapidly dissociate. And there are three subtypes of reversible inhibition, like competitive, non-competitive, uncompetitive. So I will ask you for the homework to check which type of inhibition do you see. Here is an example of a competitive inhibitor. So it binds at the active site and thus prevents the substrate from binding. These are molecules that often mimic the substrate. So it's something that looks like the substrate but is not exactly the substrate. And there are many let's say drugs that work this way as competitive inhibitors. And you see how our equations change. But here is Michaelis-Menten plot. But what's interesting is the Lineweaver-Burke plot. So you have no inhibitor, you have the straight line down here. When you have competitive inhibitor, you have straight line that goes through the same point on the y-axis, but it goes a bit upper. Why does it go a bit upper? Who might say? Why all these points are higher than points with no inhibitor? Lineweaver.
It's so
Yes.
1 divided by velocity.
Yes.
So yeah.
So it's 1 divided by velocity. So the lower the velocity, the higher is 1 divided by velocity. Yeah. So 1 over 10 is smaller than 1 over 5. So if your velocity was 10, you had 1 over 10. If your velocity becomes, with inhibitor, becomes 5, you get 1 over 5 that is higher. So that's why they go higher. So here it is. So you have a little bit another apparent value of Michaelis constant. So here is the formula, and here is the final here. Good. Another example, so-called uncompetitive inhibitor. So it binds only to the inhibitor and substrate complex. So here is the scheme. So we have enzyme, we have inhibitor, and we have substrate. When enzyme binds to the substrate, there is the probability of creating enzyme substrate inhibitor complex. So here it is. And it does not perform any catalytic activity. Only when inhibitor dissociate, catalytic activity may be performed. This is called uncompetitive inhibition. And here is example of the Lineweaver-Burk plot. Once again, all points of the inhibited version are higher than the points of the of not inhibited enzyme. But here you see these lines, they go parallel, so they are parallel, and they intersect this line, the x-axis, in different points. And here we have different, let's say 1 over Vmax, so Vmax has changed somehow. So it lowers Vmax and Km because unproductive inhibitor enzyme substrate inhibitor complex. Good. What else? Non-competitive inhibition. Here it is. So substrate can bind either to the enzyme or enzyme inhibitor complex. However, enzyme substrate inhibitor does not proceed to form a product. In pure non-competitive inhibition, Ki for the inhibitor binding to E is the same as for binding to the ES complex. So it does not differentiate between just enzyme and enzyme with substrate. So here is the scheme. You see what may happen. Inhibitor may bind to the enzyme itself, and then substrate may bind, or enzyme may bind with substrate, and then inhibitor will bind, or inhibitor will dissociate, and enzyme substrate will give you some sort of product. What's interesting for you, once again, look at the Lineweaver-Burk plot. We have, once again, two lines. Now they have the same point on the x-axis, but different points on the y-axis. So in this case, only Vmax has changed, so you have new Vmax apparent, but your Michaelis constant did not change. Okay, so when I will ask you for the homework to do something to find out what kind of inhibitor do you have by given experimental data, you basically will need to draw a Lineweaver-Burk plot and then compare which of these three types your result is the, let's say, the closest to which one of them. Okay, here are some examples for you. These are inhibitors used in medicine, and probably you at some point in your life you all have taken aspirin. So this is S-acetylsalicylic acid, and this is an irreversible covalent inhibitor that inhibits so-called cyclooxygenases. It inhibits, I guess, both. There are two types of cyclooxygenases, so COX-1 and COX-2, and I guess it blocks them both, and they are responsible for synthesizing such things as prostaglandins, and that prostaglandins, they do all different things. I don't want to get into the details right now, maybe sometimes later, maybe never. I don't know. Penicillin, so it contains a beta-lactam ring that covalently binds to transpeptidase, blocking bacterial cell wall cross-linking. So it basically inhibits synthesizing of bacterial cell wall, and so on. So many examples. Some more examples with the different inhibition types. So when we have all these uncompetitive or non-competitive, so some drugs for Alzheimer's disease or something like that. interesting part for you, there is the theory. So all that schemes that I showed you, they are basically maybe thought as a part of so-called biochemical systems theory. I will show you some examples just to show you what do we have there. Oh, I definitely forgot these this thing. You may play around with these demo. So there is a link in our slides, and you see here are different types of inhibition. So here is competitive inhibition. So you add the concentration of the inhibitor, and you look what happens. Here is the uncompetitive. Once again, you change concentration. There is here is non-competitive. So these plots may remind you which one is which. And here is example of self-inhibition, but when the enzyme is inhibited by the products it creates. That pretty often happens, but I do not want to consider this thing in lecture, so let it be. Okay, so what do I have here for you? I hope this loads pretty well, and I'll be able to show you a little bit of neat things. I'm not sure why that loads for so long. Okay, let's try exiting and reloading. Oh god. Recently all software works just awful. Here are some of my notes. I wrote them basically as I was a student. I'm not getting into any details of that. Maybe I can run all. Let's run all. Yes. I don't know, does any one of you code Python? Yes, I see. Good. So at least we have few Python coders. So these are examples. I don't know what that ligand receptor binding. Don't need that. That's Lorentz attractor. What's interesting from the biological point of view, there are some articles that describe that sort of biochemical systems, and they derive the differential equations, for example, for this sort of system like bacteria interacting and so on. You can write down that equations, you can get the constants, measure them somehow, and if you solve that equations, you will see that the concentrations of certain components will change in sort of periodic manner. So here is the plot that shows basically that periodicity. Here are some other examples of this type, some glycolytic oscillations in yeast, for example. So why not? But the interesting part I want to show you, I have created, yes, this one. This is the part of code I wrote specifically for you. That's, of course, that comes from sort of article, so it's not that I'm so smart to get this idea. So it turns out that you can create a biochemical system when you have some enzyme. These enzyme can be phosphorylated or dephosphorylated. So enzymes, they can be turned on and off, well, in a different manner. So I showed you some sort of inhibitors, but of course you can do that in different ways, and one of the most common ways is to phosphorylate or dephosphorylate the enzyme. Depending on the enzyme, that might turn it on or off, so that pretty depends, but that can be. And here you see that we have certain chemicals, we have their concentrations, and we can write differential equations that describe how these chemicals influence the amount of the enzyme we have, the dephosphorylated enzyme. And it turns out that this system is pretty, let's say, pretty interesting. So this enzyme is always synthesizing some sort of product. So here it is called the response. So this is the concentration of the product synthesized by the enzyme. And there might be some sort of what they call signal. It's some another chemical that influences these sort of mechanisms. So it just adds to this system and influences the balance between phosphorylated and dephosphorylated enzyme. And at some point I add this sort of signal. You see this blue this blue line. In the practice it should be much smaller, so you see oh, it's not blue, it should be orange. So orange line, this is the signal, and you see that response changes in as the result of this signal. In practice this signal is basically much smaller. You see here 1000 times signal, so this orange line should be almost invisible basically, but the system reacts. But what's interesting, suppose I make this signal bigger and bigger. So it just at some point I have added a little bit of some chemical and then I did nothing. So I see there is a bit of a splash in a response, and then it gets to its basic level. But if I get through third certain threshold, so you see what happens. So
An equilibrium
yeah,
happens, reaching an equilibrium.
So it's not like we're reaching, so we had equilibrium, but we have so-called bifurcation. There are two possible equilibria. The one equilibrium is down here, so low level of the response, and the other one is here, so high level of the response. And depending on the height of the signal, we can switch from one equilibrium to another. So this is called a bias switch. So I have added some signal, and if it surpasses certain threshold, the system switches and gets to a new equilibrium. So there are two equilibria to synthesize a low response or to synthesize a lot of that chemical and high response. And we have basically switched to the high response. And you see there is no more signal after these points, so it's almost zero, but this high level, it stays. So this is called a bias switch. So probably if you can make these sort of systems, and in this article there are at least a few of them that are described, you can probably come out with the idea how do you construct a computer basing on these sort of chemical reactions. So here is just a neat part for you. So here are certain links to other works if you feel interested, that for you. Okay, I guess I'm running out of time, but just a few nice videos for you. So basically I showed you I wanted. Here is this sort of reaction, but not for biological system, just a chemical system. So it is called a Briggs-Rauscher reaction, and look what happens right now. Did you see that? So this is a real deal. So it's actually how that works in practice. So this reaction is oscillating, and you see that oscillations by changes in color of the reactants. So I will not go into any of the details, Here is one more example, so-called Belousov-Zhabotinsky reaction. So here you see this reaction happens in this sort of petri dish, and you see these sort of different colors and flashes, and they basically represent how concentrations of chemicals change. So this is a real thing as well. I guess I have taken this one from the Nile Red's channel, so it's one of his most famous videos ever. Okay, not going through that. That's just some sort of representation how a different substrates binds to the enzyme. So here we have enzyme. It takes two substrates, binds them, and then they react and then go away. It may be the case that they can bind in different order and that still works. So it's called Cleland notation. It may be that enzyme binds in a sequence one substrate, then dissociates one product, then binds another substrate, dissociates another product. So nothing that. And activity regulation. So what I told you, the allosteric control. You may have multiple forms of enzymes or so-called isoenzymes, so that's a way of regulation as well. You can synthesize different enzymes. You can do reversible covalent modification of the enzyme, so you perform certain reaction and you make it more or less active. You can get so-called proteolytic activation, so some enzymes are synthesized in a form of precursors called zymogens or proenzymes, and at some point in time, these proenzymes are transformed into actual enzymes by cleaving certain bonds. And you can control the amount of the enzyme presence, that's important form that takes place in form of level of transcription. So probably if you measure that, and this is the last slide. That's just a just shows you the allosteric effects and Hill equation. So here we had, just for comparison, here we had the Michaelis-Menten equation, and here we have the Hill equation. So almost the same, you just pick up these sort of ends here. So so nothing really drastic. It is said it works well for the hemoglobin, and here you see the meaning of N. If it's greater than 1, this is a positive cooperative binding, so one ligand molecule binds and it makes the affinity for other ligand molecules bigger. So for example, hemoglobin. If it's less than 1, it's negatively cooperative binding, so ligand molecule bound, and other ligands do not want to bind. And N equals 1, it is not non-cooperative, completely independent binding. So it's basically the Michaelis-Menten kinetics for the enzyme. Good. I guess we were able to get through everything we had. If you have any comments, questions, please.
Probably I didn't understood what like there was energy total. Like
Where it was? Should I?
before the like km
Uh,
constant definition.
Km constant definition. Let me find that.
Yeah, or
This one.
this slide. Yeah.
And where should I look? K.
This equation is like y is V max divided by
This
E
one.
total. What is E total represent?
This
Is it
is
like the whole
the
system?
E is enzyme. In these brackets means the concentration of the enzyme. E total means the total concentration of the enzyme that you tried to add to your test tube. You see the thing when you add some enzyme into your test tube, some of the enzyme is free, so it's doing nothing. Some of the enzyme associates with the substrate and transforms into enzyme substrate complex. We basically don't know how much of the enzyme is in a free form and how much has transformed. But we know how much initial of the initial enzyme we have added to the test tube. And that is basically, if we add the concentrations of these two, we will get E total. So that's
Okay,
the
thanks.
yeah. So that's the total enzyme that you have added to the test tube. Then after you have added, it will start reacting. Some of that enzyme will be in form of enzyme substrate complex, some will be in free form. Of course, we don't know how much of that enzyme is in one form or another, but we know how much we have added. So that's why this E total is so interesting for us, because we know how much do we add that's measurable. I know there is one, let's say, 1 mole of the enzyme per liter, and I have added, but I don't know whether there is half a mole of the free enzyme or there is 1/4 of a mole, but I know how much I added. Yeah, maybe other questions, please. Comments? No. Good. No comments, no questions. Then have a nice evening and see you, I guess, next Tuesday.